So now we'll talk about how you typically deal with confounding factors. And so confounding factor is some kind of phenomena or set of phenomena that cause associations to appear in your GWAS study where those SNPs don't actually drive variation in the phenotype that you're interested in. And so one of the besides being able to test for association between SNPs and continuous phenotypes, this kind of line fitting procedure is really handy for being able to correct for kind of what's called confounding variables. And this is something that the Fisher's exact test that I talked about earlier in the lecture cannot do. And so the general idea here is that the power of these kind of GWAS studies really stems from the fact that the original hope is that because your SNPs, your genotypes are kind of generally determined at the time of conception, the idea is that if you can find real associations between SNPs and a phenotype, then those should be real in the sense that lots of people, for example, try to find relationships between RNA expression or chromatin accessibility and phenotypes. And even though you can find strong correlations between, say, RNA expression and some phenotype like BMI, you never know which one caused which. Because, for example, increase in BMI can lead to changes in gene expression and vice versa. And so even when you find correlations between RNA expression or accessibility and some phenotype, you don't know which caused which. But in the case of SNPs, what's kind of tantalizing about genetic association studies is that because the SNPs generally come before the phenotype, then potentially when you find associations, those are possibly causal, even though there's a very big leap of faith there in that kind of conclusion. And so one of the biggest challenges that people figured out when they first started doing these association studies is that there's lots of confounding variables. And so a confounding variable is informally some kind of variable that can explain your association between SNP and phenotype. And that basically takes away the explanatory power of your SNP. So it basically is a variable that can say, okay, I can explain why this SNP is associated with this trait. And therefore, in reality, that even though your line of best fit tells you that this SNP is correlated with some phenotype, there's some other explanation as to why this correlation exists. And so to give you a hypothetical example of when this would happen, suppose that, and I'm not claiming this is actually true, but I'm just saying that suppose that males on average had significantly higher BMI than females, and suppose that this is for reasons other than genetics, right? And so suppose that males on average just ate much more poorly than females. And so that means that on average, if you just picked a male off the street, they would tend to have higher BMI than females. Now, further, suppose that there were certain SNPs that were better represented in males versus females. So in this hypothetical case, suppose that the T allele, which was deemed the reference allele here, was basically in a higher proportion of males than it was in females. Then in that case, for this particular SNP, your association test might actually tell you that the T allele in this SNP is strongly associated with BMI. And the reason for this is that if the T allele is heavily biased towards males, just because of genetics, then there will be many more males represented in the TT genotype compared to, for example, the AA genotype. Right, and furthermore, if males have significantly higher BMI than females for reasons other than genetics, so for example, because of diet, then the TT allele individuals will have a much higher BMI than the AA individuals because males have a higher BMI than females in this hypothetical scenario. And so in that case, when you draw your line of best fit, you're going to get a positive slope. And therefore, the association test is going to tell you that the T allele is strongly associated with increases in BMI. But this is a false association because you're only seeing an association here because the T alleles are associated with being males, and being a male is associated with higher BMI. But in this specific case, being a males having a higher association with BMI is not due to genetics; it's due to diet, and so that essentially means you get a false positive here, and basically gender becomes a confounding variable. And so it turns out to be relatively straightforward to deal with confounding variables when you know what they are, right? And so in this case, if you know that gender is a confounding variable because you know that males have a higher BMI than females due to, for example, diet and not genetics, then what you can do conceptually is you can just calculate the average BMI for males. You can calculate the average BMI for females. And then essentially for every male, you can subtract off the average BMI for males. And likewise, for every female, you can subtract off the average BMI for females. And what that'll do is it'll kind of adjust the BMI value for each individual according to their gender. And then you'll get a scatter plot like what you see on the right. And now if you were to fit a line to the scatter plot on the right, where you've kind of corrected for gender-specific differences in BMI, then you'd find out that you essentially get a flat line, which again means there's no significant effect of genotype on BMI. And so you would conclude correctly that the genotype at this position is not associated with BMI. So the more typical approach to correcting for confounders is by using a technique called nested models. So the idea of a nested model is that if you want to test whether or not a SNP is significantly associated with BMI beyond what you can explain using a confounding variable like gender, the idea is that you want to basically fit two lines to your data. So the first line you fit is a line where you say, okay, my response variable BMI is equal to beta times one variable, SNP. And you add in a second variable for your confounding variable like gender. So you add plus beta 2 times gender. And so beta is the effect of your SNP and beta 2 is the effect of gender. And so you fit this model by making some estimates of the slopes beta and beta 2 that maximize your goodness of fit or equivalently minimize your squared error. And then what you do is you build a second model. You fit a second line where you just say BMI is just some function of gender. So you fit BMI equals some beta 2 times gender. And so the idea here is that the second model where you only have a gender variable fitting to BMI is nested within the first model where you have both gender and SNP. And it's called nested because the goodness of fit of the first model where you include SNP and gender should always be at least as good as the goodness of fit of your second model. The reason for this is that no matter what line you fit for the second model, no matter what you set beta 2 equal to for the second model, you can fit an equivalent line in the first model by setting beta equals to 0, by setting the effect of SNP to be 0. And so what that means is that in the worst case scenario, if the SNP has no explanatory power for BMI, then your first model should be able to explain BMI just as well as the second model because they're the same. If beta equals 0, then model 1 and 2 are the same. If the SNP has any kind of explanatory power, so if beta is anything other than 0, then your first model should have a smaller error than your second model. And so the idea of this kind of nested model approach is that you fit both model 1 and model 2, and then you check their goodness of fit. And if the goodness of fit of model 1 is about the same as model 2, then you can conclude that that particular SNP you're looking at is not really associated with BMI once you consider gender. But if the goodness of fit of model 1 is much better than the goodness of fit of model 2, then that means that the SNP added a lot more value to your model than beyond what gender can add. And so that means that the SNP is associated with BMI when you condition out gender. And so in that case, then your p-value that you would calculate would be small. And so genetic association studies have a lot of challenges. And so the first challenge they run into is what's known as winner's curse. And so the idea here is that when you perform a genome-wide association study, what you're doing is you're repeating the test, this line fitting test that I just talked about on the previous slide, on a large scale. Like typically you're testing like anywhere between like 600,000 and 10 million SNPs for association with a given phenotype. And the problem is that typically when you perform a genome-wide association test, you are testing, say, your 10 million SNPs, and then you're typically ranking them by their p-value. So you're looking at either the SNPs with the smallest p-value, because that's this, most statistically significant SNPs, or you're ranking them by effect size, and you're looking at the SNPs that had the largest estimated effect on the phenotype per copy of the reference allele. And so genome-wide association studies, GWAS, basically run into the problem of winner's curse, where winner's curse just means that the SNPs with the smallest p-value or the largest effect size tend to have their effects overestimated. So the p-values are smaller than they actually should be, and the biggest effect sizes are bigger than, they're larger than they actually are in the real population. And so one way to understand winner's curse, the typical explanation for winner's curse is through the idea, it's typically explained through the idea of auctioning, right? And suppose that you're running a blind auction in which, your auctioneer is basically putting up an item for sale, and anyone can bid for that item. Everyone writes down a number, what they think it's worth to them, submits the bid, and then basically the person who offered the most basically wins the auction and can buy the item. And so in a typical blind auction like this, what you'll notice is that even if everyone is bidding for an item at approximately the actual value of the item, right? So suppose that you have like 100 people bidding for like a car, and suppose everyone is equally good at estimating the value of the car, so everyone bids about the cost of the car, or the true value of the car. Because the winner of the auction is the person who bid the most, that means that the car will actually tend to sell for more than it's actually worth. Because if everyone's bidding about the value of the car, because there's always some kind of random stochasticity there's some random variation there. Even if everyone's bidding about the value of the car, there's going to be some bidding higher than others. And because the person who bid the most wins the auction, then the car will sell for more than it's actually worth. And so it's a similar principle with any kind of large-scale hypothesis testing, in this case GWAS, because if you're only looking at the SNPs with the smallest p-value, because there's noise in how those p-values are estimated, in part because your sample sizes are typically small, then that means that your p-values are they're essentially overrated. And so the other problem that GWAS tends to have is a problem called hidden confounders. So I spoke previously about known confounders like gender, where you might know ahead of time that certain genders have higher BMI than others, and those are easy to deal with. But the harder problems are confounding due to things that you don't know about, right? And so one of the common kind of hidden confounders that people have to deal with when they do genetic association tests is basically genetic similarity. And so the idea here is that genetic association studies essentially basically look for correlations between genetic similarity and phenotypic similarity. So the idea of association studies is that if two people who, if you have a case where the more genetically similar two people are, the more phenotypically similar they are, and that essentially suggests that genetics explains phenotype. And so that's how association tests are generally, that's how they generally work, even the line fitting one we talked about. And so the idea with hidden confounders is that if genetic similarity co-occurs with phenotypic similarity for reasons other than, genetics explaining phenotypes, then that's a problem. And so to give you a few examples, suppose that you're doing an association study where you're studying families of individuals. So here you have a problem, and suppose furthermore that you're looking at a phenotype like BMI, like obesity, right? So the problem you have here is that if you don't consider the fact that you have some individuals who live together, so individuals within a family, and some pairs of individuals who do not live together or are from different families, part of the problem you run into here is that individuals within a family obviously typically share genetics, right? Kids share a lot of genetics with their parents. So basically people within the same family tend to share genetics, but they also tend to have certain shared phenotypes like BMI, because, for example, people that are living together in the same nuclear family might tend to eat together a lot because the parents, for example, cook or buy out for the kids. Then you can have groups of individuals where they share similar genetics. They also share similar phenotypes because everyone's eating the same thing, and so maybe they have similar BMIs. And so it might look like genetic similarity is correlated with phenotypic similarity, but it's actually not driven by genetics. These people within the same family don't have the same phenotype because they share genetics. They have similar phenotypes because they're eating the same thing. And so in this case, again, diet is a confounder. And so if you don't know what the diets of the individuals are that are in your study, then you can't correct for it. And so it's a hidden confounder. Similarly, problems can arise. So again, number one, problems arise at kind of like the family relatedness level. Problems can also rise at a different level of genetic similarity. So consider similarity due to ethnicity, right? And so broadly speaking, African individuals look more similar to other African individuals than they do to European individuals. And similarly, Asian people tend to look more similar genetic-wise to other Asians than they do to Europeans, right? And so ethnic diversity, or essentially ethnic similarity, can also be a confounding factor. Because suppose, for example, so the example I always like to give is one of stomach cancer, right? And so if you were to, for example, suppose you were to do an association study of stomach cancer, and you naively walked out on the streets of Davis and you basically tried to genotype the first 100 people you found that didn't have stomach cancer, and then genotype the first 100 people that you found that did have stomach cancer, and then you went and did an association study. If you didn't consider ethnicity, you might get a lot of signal. You might find a lot of genetic signal in your study, but that genetic signal may not be real because, for example, we know that in certain Asian cultures, people might tend to eat a lot of pickled food or preserved food. And so the problem you have there is that it's kind of a well-known association that the more kind of preserved or pickled food you eat, kind of the higher the incidence of stomach cancer you have. And that's primarily thought to be because the chemicals within pickled foods tend to predispose you to stomach cancer, right? And so then the problem you have is that if you do this naive genetic study where you don't consider ethnicity, then what you'll end up having is the case where your controls have, if you're living in Davis, your controls would be overrepresented in, say, European individuals, but your cases would, it might be, for example, overrepresented in Asian individuals. And so when you do this association study, if there's any SNPs that are strongly associated with ethnicity. And so if there's some SNPs that are, if they're only prevalent in Asians and not Europeans or vice versa, then those SNPs will look like they're associated with stomach cancer, even though, by and large, they aren't actually. And so these hidden confounders basically cause spurious results. So you get SNPs, you'll get false positives, and you'll get SNPs looking like they're associated with a phenotype when they're actually not. And you also end up with lack of power in the sense that sometimes when you don't correct for these hidden confounders, you actually lose power to detect real SNPs. And so, just to harp on this point further, underlying a lot of these methods like line fitting is a fundamental assumption that, when you're fitting lines, for example, you're assuming that each individual represented by a point is kind of sampled what's called identically and independently from each other. And so essentially the idea is that no pair of individuals is more related than other pairs on average, right? And so this assumption is broken when you have studies in which there are families within your cohorts, or broadly speaking, you have different representations of different ethnicities. And so those are biological reasons why this assumption that people are what are called IID, or independent and identically distributed, can break. It can also break if you have what's called batch effects. And so a batch effect is, I believe we talked about this in previous lectures, but a batch effect is an observation that samples that, for example, get sequenced at the same time tend to look more similar to each other than samples sequenced during different times. And so batch effects can also drive a lot of hidden confounding variation as well. And so just to harp on the point again about problems related to, for example, systematic differences in cases versus controls in terms of ethnicity, if your cases have a significantly different proportion of one ethnicity compared to the controls, suppose your cases and controls have two populations, and suppose the proportion of people who are from population 1 or 2 differ in cases and controls, then what that means is that any SNP that distinguishes population 1 and 2 will also distinguish cases and controls, and therefore will also look associated in your GWAS study.